Okay, welcome to Let's Get to the Marks. I'm going through the AQA GCSE Combined Science Trilogy, Biology, Higher Tier, Paper 1, 2018. Let's get into it. You need to revise, 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 do as many papers as you can and smash those exams. Yep. Okay, I recommend that you grab a scrap bit of paper, grab a calculator, grab a pen or pencil. Don't matter if it's a felt tip or a color or whatever. Just get some scrap paper. Pause the video at the start of each question, have a go, then unpause and watch my explanation. Okay, guys, let's get into it. Let's go smash those GCSEs. Question one. This question is about cell structures. Draw one line, 1.1, from each cell structure to the type of cell where the structure is found, and it's worth 2 marks. So cell structure, we've got nucleus. Where do we find the nucleus? Do we find them in prokaryotic cells? No, that's like bacteria. They don't have a nucleus. Plant cells only? No, animal cells do have them too, and fungi cells and protist cells. So there's 4 types of different cells that we call eukaryotic cells. All of them have a nucleus. They are fungi, protist, plant, and animal. 4 of the kingdoms. Right, we've got permanent vacuole. Which of these cells have a permanent vacuole only? And that is plant cells. Yes, bacteria can have a vacuole, and even animal cells, white blood cells, temporarily have a vacuole when they engulf other things. So we call it a permanent vacuole, and that's found in plant cells only. Plasmids, yep, they're little tiny circles of DNA. They are found in prokaryotic cells. How many marks did you get? Check it off. If you don't know the answers to these, grab a revision card, write down some questions like what doesn't have a nucleus, what has a nucleus, blah, 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 make some up. All right, let's go, let's go, let's go. Enough of my whiffle, waffle, waffle. Let's get on, let's smash those exams. Let's get as much revision as we can done. So, shrinking this so it fits on the page. 1.2. Figure 1 shows a plant cell. Here it is. It's got A, B, and C. What are the structures A, B, and C? Let's look at A first of all. What is this very, very, very large structure in the middle of a plant cell? Now, some people may make the mistake of saying nucleus, so they may go for this answer here. But of course, this is the permanent vacuole. So our answer here has to be one of the bottom 2, as that's the permanent vacuole. What's B? Well, it's the smallest thing that you find inside a cell, and that is a ribosome. They're the smallest organelle in the cell, really. So we've got vacuole, ribosome, and then, of course, C is the cell wall, which is the outer layer of plant or fungi cells, and bacteria cells for that matter, have cell walls. So there we go. Boom. Another mark. Right, let's go, let's go, let's go. Ba, 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 ba. What we got going on here? A student observes slides of onion cells using a microscope. Figure 2 shows 2 slides the student observes. So you can see A here looks like the cells are really quite large compared to B, but they're yeah, a little bit blurry. What are these black circles? You can see these little black dots in B. The cells on the slides are not clear to see, so we can't really make out the cells very clearly. Describe how the student should adjust the microscope to see the cells on slide A more clearly. So they've obviously used the rough focus. You've got 2 knobs on your microscope. You've got a rough focus and a fine focus. And you turn the rough focus until, or the coarse adjustment knob, it's also known as, until you roughly get the cell in view like this. And then when you want to see the detail, you then hit the smaller knob and you turn that, and it's called the fine focus. So adjust the fine focus. You can use the word knob if you like, if you get excited about that. But anywho, let's move on. 1 mark. Right, let's go. Okay, 1.4. Describe how the student should adjust the microscope to see the cells on slide B more clearly. So we can see the cells on slide B. We've got a little problem here, is that they're just not big enough. So what do we need to change on this? We need to select a higher magnification. And we're going to do that on the objective lens. Now, hopefully you can see from my crusty little diagram here, the objective lens is that little turny thing with the 3 cylinders on it. And you've got like times 40, times 500, times 1,000, whatever. So on the objective lens, the eyepiece lens is the one you look through. That doesn't change the size. It's the objective lens that we can adjust on the fly. Okay, that will get you 2 marks, one for select a higher magnification, and then the other one, using the objective lens. Right, we're on to 1.5. I'm just going to shrink that down so you can see this is a 3 mark question, total of 9 marks for this first question on this paper. The student made the necessary adjustments to get a clear image. Figure 3 shows the student's drawing of one of the cells. So we've got figure point 3 here. You can see we've got a cell here. Just be thinking to yourself, what type of cell is that? We've got a nucleus, we've got some cytoplasm and a cell membrane. Anyway, you don't need to know that for the answer to this question, but it's worth thinking about when you see these diagrams. Okay, the real length of the cell was 280 micrometers. Calculate the magnification of the drawing. So here we've got 112 millimeters. That is the image of the cell. Obviously, the cell is not going to be like 11 centimeters long. There are some cells in South America that get that big. Some amoebas they found that are actually quite big and visible with the naked eye. But that's very unusual. Normally you need a microscope. So anyway, what am I waffling about? You need to know this triangle. You've got to learn it off by heart. I call it I am. However, in AQA, they quite often say real size instead of actual. But the way this goes is you've got image, okay? Oh my gosh, I can't spell image. That is pretty damn bad. We got image. So the image size is what you see when you put your eye to the microscope, and you have that over the actual size, which is going to be really, 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 really small. Because these things we're looking at with microscopes are tiny. They're called microorganisms. And then we have the M stands for magnification. And if you can remember that as I am, then you'll always be able to work these out. So it is 3 marks, and we have got to take into effect a conversion because the units don't match up. We've got millimeters and micrometers, but I'm going to show you how to get 2 marks without even doing that. So first of all, that's the image. So if you did 112, which is the image, I, over the actual size, and they've called this the real length. Real is the same as actual, and that's 280. So if you did that and you write your answer down here, you're going to get 2 marks. But really what we need to do is convert this, because this is in millimeters. We need to put these into the same size, and this is micrometers. Now you've got a choice. We can either, to put this into millimeters, we can divide 280 by 1,000, or I can say I want to put my millimeters into micrometers, and then I can times this by 1,000. I find it easier to turn millimeters to micrometers, and you do that by timesing by 1,000. So it's actually 112,000 micrometers over the top. So we've turned the millimeters into micrometers, and we're going to divide that by 280. And this is going to give us our magnification. Now, your magnification, it's normally a whole number, so you'll kind of know you've gone wrong if it isn't normally, but there we go. And it equals 400, and that makes sense. If you look down a light microscope on the objective lens, you might get 200, you might get 50, you might get 1,500 max, but there you go. Right, anywho, let's do the marks. So 1 mark for rearranging the equation. So if you just put image over actual, or you can call this I over A, or you could have said I over R, the same thing. So if you just worked out that you need to divide the image by the actual, that will get you a mark. Your answer, whatever it was from doing that, will get you a mark. And if you remembered the conversion, that's the bonus mark there. Anyhow, that's the end of the first question. Let's move on. Okay, where am I? Where am I? Where am I? Question 2, coronary heart disease, CHD, is a non-communicable disease. CHD is caused when fatty material builds up inside your coronary arteries. Worth 2 marks. What is a non-communicable disease? What does communicating mean? Think of the word communicate. It means like when you talk to someone, when you engage with someone. So if it's non-communicable, this disease is not passed on between people. Communicable diseases are things like the common cold, the flu, HIV, anything that you can pass via microorganisms to someone else. Non-communicable diseases, they can be affected by microorganisms, your chance of catching them, but actually they're not able to pass on to someone else. So you're thinking about things like diabetes, you can't catch that off someone. Cancer, you can't catch that off someone. Heart disease, you can't catch that off someone. These are non-communicable. So what we say are diseases, whoops, yeah, that was all right. Diseases that cannot spread from person to person, not directly caused by microorganisms. And I might give an example here because I want to get all 2 marks, and I'm not sure whether the 2 marks are for diseases that can't spread, and microorganisms might be the 2 marks, or they might want an example. So I'm going to say like cancer or diabetes, just so the examiner knows I know exactly what I'm on about. And the 2 marks really for this is bum bada bing bada bang bada bong, cannot be spread, and by microorganisms were really the main 2 marks on the mark scheme. But I like to give an example as well. So there we go. Right, let's zing down. Okay, figure 4 shows a coronary artery of someone with coronary heart disease. So we've got the artery wall, we've got this fatty material inside. Explain how CHD can cause a heart attack, and it's worth 3 marks. Do you remember what the space inside any blood vessel or any tube in your body is called? It's known as a lumen. Anyhow, just thought I'd mention that. Right, explain how coronary heart disease can cause a heart attack. So if we have a fatty material here, what's it going to do to the amount of blood flowing to your heart muscle? So it's going to reduce blood flow. So we're going to say fatty deposits. I could have just said the word materials, it's there. Fatty deposits reduce blood flow to the heart muscle. Now, it's important really to use the word muscle. I guess reduction in blood flow would definitely get you a mark. What does this mean? If your heart muscle cannot get blood, what's it going to get less of? The cells, the muscle cells, this is, get less oxygen. So the cells are going to get less oxygen. What do we need oxygen for? Every living cell needs oxygen. What does it need it for? The cells get less oxygen, so reduce. That was a bit of a crap word. Reduced respiration or no respiration, lack of respiration, which means no energy or less energy for muscle contraction, um, of heart. So yeah, less energy for the heart to pump. You could say all those kind of things. What do you really get the marks for here? You get the marks for saying the word reduced blood flow, less oxygen, less energy. That would be 3 marks, or less contractions, those kind of things, or less pumping of the heart. Brilliant. 2.3, what we got going on here? It's a 6 marker. What? Explain how lifestyle and medical risk factors increase the chance of developing CHD. So there's a key thing here. We have mentioned two, don't know what's going on there. Right. We've mentioned 2 things here: lifestyle and medical risk factors. So what we're looking at really is we need to include both of these if we want 6 marks. If I only talk about one, I'm probably going to max out at 3 to 4 marks. So I need to talk about both if I want to attempt to get 6 marks. And then the word explain. I always say the word explain means use your science knowledge. So we want to talk about how lifestyle factors can increase the chance. And so just think about some of the lifestyle factors, what will increase your chance of developing heart disease. So we might say something like obesity, means lack of exercise, like you're not going to be able to exercise as much. And how is that going to worsen things? So we might want to talk about high cholesterol. So if you're obese, it's likely that you don't do a lot of exercise and likely that you have high cholesterol, causes fatty deposits in arteries. So that's a lifestyle factor I've just talked about there. Then we've got something like smoking. What else can we say? Let's do a little comma. Again, I'm mentioning lack of exercise. And let's say high salt diet. So what I've done here, I'm linking these. What do you think smoking, a lack of exercise, and lots of salt do to your blood pressure? These increase your blood pressure. I could have said stress as well. When you get stressed, when your parents get stressed, when your teacher gets stressed and shouts at you, any increase in your blood pressure is going to damage the walls of the artery. So it damages walls of artery, increases chance of fatty deposits. So anything that increases fat sticking to the walls of your artery, so either your diet is high in fat or high in cholesterol, or you've increased your blood pressure, will cause cracks and roughening of the artery wall. This will make fat stick to the sides of the wall and mash up your arteries. Now, if you're interested in looking at a little video on that, I got one here. So check out the link to that video, which is going to give you a nice visual, a demonstration of fatty deposits building up. And it's only a real quick video. All right. Let's move on. We've done enough there, really. I could talk about some other things like stress, blah, 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 blah. Now we want to talk about medical risk factors. So this is my lifestyle factors ticked off. all of these are choices. Like smoking, that's a choice. Going for exercise is a choice. Putting lots of salt in your diet, that's a choice. I could have been, now we want to talk about medical risk factors. So I might say diabetes, or I could say diddly dee, diddly da, diddly da, genetics. Um, yeah, actually, let's just say genetics. This is a medical risk factor, could lead to you having, so it might not be the fact that you've taken salt or that you smoke. You might be really healthy in your diet, but genetics could lead to you having high blood pressure, leading to increased chance um, of fatty deposits, or we can say plaque buildup, or atheroma, or thrombosis, all of that kind of stuff. So it could be down to your genetics. Um, Maybe you inherited diabetes, so maybe it runs in your family and you've got diabetes. That would be a medical risk factor. Um, and then like certain drugs or medications people take might also increase cholesterol buildup or high blood pressure. So most of this is linked, most of these risk factors, whether they're medical or lifestyle, are linked to high blood pressure, which is why when you go to the doctors and do a health check, they're really interested in checking your blood pressure. And so there we go. We've kind of mentioned it, we've given examples, and there we go. Yeah, if you inherit diabetes, you're more likely to end up obese as well. All of those kind of things. Right. Let's smash on to the next question. Right, we're on question three, and this question's about photosynthesis. What is the correct balance equation for photosynthesis? So, what I encourage people to think about when they're on a question like this about photosynthesis is just to double check in your head, what does a plant take in? So, we want to think about to ourselves, what does a plant take in? Just remind yourself, what does it take in? What goes into a plant? And hopefully you remember, oh, plants don't take in oxygen. We're always kind of taught that. What do plants need? They need carbon dioxide and they need water, and they make glucose and oxygen. So this is the correct equation for photosynthesis. And hopefully you recognize there are three big fat sixes in it as well. And that is the correct equation for photosynthesis. If you reverse it, it's respiration. So people will probably either get that one, go for the top one or the bottom one, because the top one is also correct, but that would be for respiration. Right, what type of reaction is photosynthesis? So I want you to think about this. We've got a leaf. Oh my days, look how bad that drawing is. Geez, no wonder I've got D and GCSE art. So we've got a leaf and we've got sunlight zinging down into the leaf. So think about sunlight. That is heat. That is energy. It's like light energy. It's heat. And that's coming down, going in. Which of these words means energy going in? And the answer is endothermic, means energy in. Exo means energy out. So respiration, which is what you're probably doing right now, everybody's doing to keep alive. Put your hand under your armpit right now, right under your armpit. You can feel heat going into your hand from your body. That's respiration. It's exothermic. It releases heat. Photosynthesis is actually endothermic. So plants actually cooling in nature because they absorb energy into their leaves. Okay, coolio, coolio, coolio. Right, let's move on. Ba, 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 ba. Right. A student investigated the effect of light intensity on the rate of photosynthesis. Figure 5 shows the apparatus used. We've got an LED light source here, so it doesn't get hot. That's pretty good for controlling temperature there. We've got pondweed with a 0.2% sodium hydrogen carbonate solution. You should know that that produces carbon dioxide. That's why they've put that in there. And we've got the pondweed under the water. Why is pondweed really good? If you use a normal plant that doesn't live under the water, you can't count bubbles. So sodium hydrogen carbonate solution releases carbon dioxide gas for the pondweed. This should also trigger you to go back and check your photosynthesis question. Now you know that pondweed needs carbon dioxide gas. This is the method used. One, place the pondweed at 5 centimeters from the light source. We can see that there. Measure the rate of photosynthesis by counting 1, 2, 3. The number of bubbles produced in 1 minute. Repeat with the pondweed at 10 centimeters. So we're going to move it there. And then at 20 centimeters. So is it going to get more light the further we move it away, or less light? Think about that. Counting the number of bubbles produced in 1 minute is not an accurate way to measure the rate of photosynthesis. Suggests two ways that we can actually measure the rate of photosynthesis more accurately. So if we want to measure the rate of photosynthesis more accurately, you might know that bubbles are pretty rubbish, right? Because what is it if I draw these bubbles? Why are counting bubbles not very accurate? Well, here's a bubble. Now, is that bubble, the fact that one plant gives off that bubble and another plant gives off these three bubbles, which one is photosynthesizing more? We can't really tell because the bubbles are all different sizes. So you might say, oh, that gave off 3 bubbles, but look, all 3 added together aren't as big as that bubble. So it doesn't take into account different sizes of bubbles. So we need a special piece of equipment. And what we do is we put a bung in here, and you've probably seen this before. It's called a gas syringe. And if we connect a gas syringe, we will be able to measure directly the volume of oxygen given off. And that's much more accurate than counting bubbles, which can be different sizes. So we can use a gas syringe. You could also use a measuring cylinder filled with water and turned upside down, which is what you do in school a lot of the time to measure this. So I'm going to say measure the volume using gas. It didn't actually ask us what equipment to use, so I think just measure the volume would be fine. Measure the volume using gas syringe or measuring cylinder. There we go. Coolio. Measure the volume, I should probably say, of oxygen, just to make sure the examiner knows I know what it is we're measuring the volume of. Right, and number 2, it's probably a little bit harder, but they've said the word rates. If you want to measure the rate, what haven't they done here? We probably need to oh, they have done it. They did record it over a minute, but in a minute you might make there's more errors. If you do it over a longer period of time, then any random error is going to have less of an effect on your results. So you're going to get closer to the true value. So over a longer length of time, e.g. 5 minutes. So if you just think about you were counting bubbles, if you count bubbles in a minute, you might make a mistake with 1 bubble and it'll affect your results. Maybe you only counted 10 bubbles and you've made a mistake by counting an extra bubble. That's a 10% error, 1/10, right? But if you counted for 5 minutes and you counted 50 bubbles and you've miscounted by one bubble, you've done 1/50. It's a much smaller error. So it's always more accurate to use larger volumes of stuff that you're measuring, or larger lengths or longer times. So always think bigger to increase accuracy. Right, enough waffle. Let's move on. Right, we're on 3.4. The LED light source does not get hot. Explain why it's important the pond weed remains at a constant temp. Right, what do we know about temperature? Hopefully you know this. Temperature is a limiting factor of photosynthesis, so we need to control it because we were changing the light intensity. Temperature is a limiting factor, so I'm just going to smash that down there. So it's going to affect the results. How would it affect the results? Because it says explain. The moment you see the word explain, think about temperature. 2 ways you can go down this answer. So you might go increasing temperature means more kinetic energy. So you could go down this route. That's going to mean more kinetic energy, more collisions, enzymes work better, faster rate of reaction. So we're going to get more successful collisions, da-di-da-di-da, bit of chemistry there. Personally, that was going to get you 2 marks, right? Personally, I'd prefer to go down this route just because when you think of things getting hot and enzymes, what do you think of? Enzymes could denature, no longer active site bind with substrate. So I've just explained you might mess it up because the enzymes might denature and the active site won't be able to combine with the substrate. So that's an alternative there. You could go down. You could say the whole lot. It's up to you, really. Okay. But what are we on? 3.5. Let's go. Light intensity can be calculated. Bosh. I think I've got a video on this. I've already got a video on this. I've covered this in another video, but there we go. Light intensity can be calculated using the inverse square law. Light intensity can be calculated. Bosh. I think I've got a video on this. I've already got a video on this. I've covered this in another video, but there we go. Light intensity can be calculated using the inverse square law, where this L here stands for light intensity, and then this is equal to 1 over the distance squared, where L is light intensity, D is the distance of the pond weed from the light source. The student placed the pond weed at 5, 10, and 20 centimeters away from the light source. Explain how light intensity changes as the distance of the pond weed from the light source is doubled. Now, it gives you the clue, everything you need to know. You must, in bold, include calculations in your answer. You get all the marks from the calculations. So let's do it. Right, let's work out the light intensity at 5 centimeters. So we're going to go, as I say, I've got a video on this already. Probably better than what I'm going to do now, but light intensity at 5 centimeters is equal to 1 divided by the distance squared. Well, the distance is 5 centimeters. So we just got to square it. What is 1 over 5 squared? It's 1/25. Now you can press equals on your calculator. You can get that in a decimal if you really, really want. But it's going to be really clear here that at 5 centimeters, it's 1 over 25. Now let's see what happens when you double the light intensity. Sorry, when you double the distance. You don't double the light intensity, that's for sure. Um, in Now you probably only need to use two of these. You could use all three if you really want, but you probably only need, well, you do, I know from the mark scheme, only need two of these. So light intensity at 10 centimeters is equal to 1 over 10 squared. What's that when we square 10? It's 100. So the light intensity has changed. What's happened to it? It's got 4 times weaker. It's gone from 1 over 25 to 100th. So it's quartered. We can say light intensity has quartered. So it's got 4 times weaker, right? Well, what happened to the distance? Distance was doubled. So as you double the distance, it doesn't get half weaker, it gets 4 times weaker. So it's like to the power of 2. So yeah, so we can say as you double distance, the light intensity quarters, right? Guess what do you think will happen as you triple the distance? Now you don't need to say this. We've just given the answer there. You're going to get all 3 marks for this. You've included calculations and you've made your statement. You're going to get all the marks. But check this out, right? Let's go from at 5 centimeters. 1 over 5 squared is the light intensity. So what we're going to do, the light intensity is actually 1 over 25. That's the light intensity. Let's look at it when we quadruple it up. What's going to happen when you quad it? So when you quad it to 20 centimeters, you're going to get 1 over 20 squared. What's 20 times 20? It's 400, isn't it? So we're going to get 1 over 400. So actually, it's got when the distance goes 4 times, the light intensity is 4 times 4 is 16 times weaker. So actually, when you double it, it gets 4 times weaker. If you triple it, it's 3 times 3, it's 9 times weaker. If you quadruple the distance, the light intensity gets 4 times 4, 16 times weaker. Bosh. Right, let's move on. I've actually got a video already on this that you might find more useful. I might have done it a little bit more slickly. But there we go, as I'm just going through this off the dome. 3.6, the students' results are in table 1. Distance of the pond weed from the light source, 5 centimeters. Number of bubbles produced in 1 minute is 129, 10 centimeters, 31, 28. Predict how many bubbles of gas would be produced in 1 minute if the pond weed was placed 40 centimeters. So they're saying if we go to 40 centimeters, so if they double the distance, what happens to this? What did we say when you double the distance? So when distance is doubled, the light intensity, so photosynthesis is going to get the actual light reaching a plant is going to get 4 times weaker. So what do we need to predict? 4 times weaker than 8 is 2. So what we want to say is 2 bubbles, and that is going to get us a mark. I predict it's going to be 2 bubbles. As you double distance, light intensity quarters, or 4 times weaker. There we go. We've just stated the old inverse square law. All right, let's move on. Hasn't come up. Inverse square law hasn't come up in a while, and this is 2018. So you might want to make sure you revise that for your exams because it hasn't come up in a while. 3.7, describe how the student could change the method to investigate the effect of the carbon dioxide concentration on the rate of photosynthesis. You should include how to change the independent variable to control variables. Look, there's 3 limiting factors in photosynthesis. So rack your brains. Can you think of the 3 limiting factors? So we've got 3 limiting factors in photosynthesis. You've got temp, you've got CO2, and you've got light intensity, right? They're the 3 limiting factors. If you change one, you control the other 2. So it's saying it wants us to change the carbon dioxide concentration. Now, they gave you a big fat clue, and I talked about it at the start. It's called sodium hydrogen carbonate. If you don't memorize that, well, if you don't, I can't even speak properly, but basically that's how we control the CO2. So we can say change, if you can spell properly, change the mass of sodium. Oh my days, what's going on with spelling? Sodium hydrogen carbonate. So you might want to do it 5 times. So like 1 gram, 2 gram, 3 gram, 4 gram, 5 gram. You get the idea. So we're going to do 5 different masses. However, keep the temperature the same. Now, what I want to do is include how you control those variables. So it says you should include how to change the independent variable. We've said that. You change the mass of hydrogen carbonate, 1 gram, 2 gram, 3 gram, 4 gram, 5 gram. However, keep the temperature the same. Do you know how you keep the temp the same? use, and you need to know this off by heart, a water bath. Very rarely is there an exam that goes past, or do a set of GCSE science exams go past, where either in chemistry, biology, or physics, you don't have to talk about maintaining the temperature using a water bath. It's 1 mark every year. It comes up on one of them. So keep the temperature the same, use a water bath. And then I'd say the light intensity, we need to keep the light in sugar intensity the same. Now, how, I hate it when that does that. That's that weird thing you get on Microsoft, right? Intensity the same. Keep the light intensity the same. How do we do that? By using a ruler to uh, keep distance of lamp same every time. So you might do like, I don't know, e.g. 10 centimeters. You're going to put the lamp at 10 centimeters and not move it. So you always get the same light intensity. Keep the temp the same by using a water bath. You might say LED light. That's what they talked about, but use a water bath. And that's going to get you 3 marks. Bosh. Okay, we're on to question 4. Zing, ding, ding. Let's go. A student investigated the effect of different concentrations of sugar on pieces of carrot. Bosh, bish, bash, bish, bish. I sense osmosis. This is the method used. Weigh 5 pieces of carrot. Place each into a tube. Sorry. 3. Into each tube, add 20 centimeter cubes of water and one of the sugar solutions as shown in figure 6. Leave the apparatus for 2 hours. Remove the carrot and dry each piece on a paper towel. Reweigh each piece. Calculate the percentage change in mass of each piece. So this is going to be on photosynthesis. On osmosis. Figure 6 shows how the investigation was set up. So we've got one bit of carrot that's been dumped into pure water. Look, it's got 0.0 mole per decimeter. That's concentration. So concentration of 0. Then we've got sugar at a concentration of 0.2, sugar at 0.4, sugar at 0.6, and sugar at 0.8. Here's the concentrations, and here's the change in the mass. Suggest why the student calculated percentage change in mass of each piece of carrot, 1 mark. So when you start off and you cut all your carrots, you make sure they're the exact same length, exact same width, the same size. However, what you can't control is the number of cells, the density of the cells, how much glucose, carbohydrate, protein is in each cell. So you actually can't keep, you could cut 2 bits of carrot absolutely identical in shape and size. They're going to have different masses because you can't control the internal contents of the cells, which means the carrots will have different starting masses. So we need to do the percentage change rather than the change in grams because they're all starting at a different mass. So it's easier to compare. You can always just say easier to compare. That is a great thing. And then I would say because the carrots have different starting masses. So both of these would, any one of these 2 statements would get you the marks. I like to put both down. So it's easier to compare using percentages, and both the carrots have different starting masses. So there we go. Oh no! I hate doing graphs on my videos, but unfortunately, you're always going to have to do these dirty graphs. Okay, so when doing this, peoples, the first column here, the concentration of sugar solution, is always the independent variable in every table, and that goes along the x-axis. They've already labeled that for us. So we're going to get a mark for labeling the y-axis with the next column, which is percentage change in mass. So we've got percentage change in mass, and that is going to get us one of our marks. We then need to plot these points. So I'm going to go 0, 0, which is right on this line, and go up to plus 24. So I need to make a scale. So a suitable scale is going to get me marks. So this scale, the maximum is 24. So I'm going to go 5, 10, 15, 20, 25. Now I need to work out how much each square, well, there's 10 squares that cover 5 on my values on the side. So each square is worth a half. How do I know that? Well, I go 5 because this goes from 20 to 25, and I divide it by the number of small squares. So 5 over 10 equals 0.5. Each baby square is worth 0.5. So let's do 0. I want to go up to 24. So let's go 21, 22, 23, and 24 is here. So that is 24. There's my first mark. Now, 0.2 is 12. So I come to this line here, 0.2, and I go up to 12. So remember, that's 1. That is 2. So that there is 12. Then I go 0.4 and plus 1. So 0.4, and we're just going to 1. So just 2 squares up. Remember, each baby square is worth 0.5. 0.6, we're on a minus number. So what I want to do is minus 5, minus 10, minus 15. I've only just fit this in. Boy, I got a squeeze there. 0.6 is minus 8. So we're going to go minus 6, minus 7, minus 8 is here. And then at 0.8, we've got minus 15. So I'm here. Okay, now I need to do a line of best fit. Sometimes these are like a curve, but we're going to bust out a straight line on this one. This looks very straightish. It's kind of a curve, isn't it? Estimate the conc of the sugar inside the carrot cells. Use your completed graph. What you're looking for here, guys, how you do this on every osmosis graph, it's where the line that you've drawn, the line of best fit, crosses the x-axis. That is the concentration. So I'm really looking here. What is the scale here? So that's zero point 0.2 and I've got 10 squares. So each square is worth 0.02. That's a bit crazy, isn't it? So that's like 0.5 is there. So in between 0.4 and 0.5. So you're looking at like 0.45, something like that. So 0.42, 0.44, 0.46. I'm going to go for 0.46 according to my graph. Although, to be honest with you, it yeah, could be like 0.43 or something. I mean, how accurate my graph is, I don't know. But there we go. That's going to get you the mark. Explain why the mass of the carrot in the zero point 0.6 molar decimeter sugar solution changed. So what we're saying here is, as I scroll down there and I rub that 0.46 out, this is worth a fat 4 marks. So check this out, peeps. This is worth a fat 4 marks. Explain why the mass of the carrot in the 0.4 explain why the mass of the carrot in the zero point 0.6 mole per decimeter sugar solution changed. So why, when we were at 0.6, did the carrot lose mass? So that's the first thing we want to say. And look at the word explain. We need to use science, but we're going to use a description to get the first mark. So the carrot lost mass. So that is going to get us a mark. Why did it lose mass? Just think about it. What must be happening if a carrot has shrunk? This is the osmosis practical. What has happened if the carrot has shrunk? What has come out of it? Why has it lost mass? So it has lost water, which has moved out, right? That's going to get us a mark. By osmosis, oh, might get us a mark, from a high water potential or concentration to a low water concentration into the solution. So I'd probably like to say the water has moved out of the carrot, which we've said the carrot has lost mass because water has moved out by osmosis from high to low. I could have whacked in semi or partially permeable membrane there, as that's part of my osmosis definition as well. That would have been quite nice to bust that out as well, the old osmosis definition. But here we go. That's going to get us 4 marks. So let me show you where the marks are on the mark scheme. So saying the carrot lost mass is a mark, fairly obvious, right? Saying it was caused by osmosis, pretty cool, right? Saying it was going from a high to low is the third. And the one that I missed out that I did mention verbally was I should have added across a partially, you can say semi, um, permeable membrane. So that is the fourth mark. So really, you just need to know your definition of osmosis. Osmosis is when water moves from a high concentration to a low concentration across a partially permeable membrane, and that was 3 of the marks. The carrot lost mass because water moved out across a partially permeable membrane by osmosis from a high to low. Bosh. Right, what is the next questione? Let's go. I'm still keeping the graph up there in case I need it. I haven't really checked through this paper before doing it. The student repeated the investigation using boiled pieces of carrot. They didn't change in mass. Suggest why? Right. Well, boiling a carrot is going to basically damage the cell membrane and actually melt the lipids that we find in the cell membrane, the cholesterol and lipids. So damages the cell membrane, and that will get us a mark. It actually will melt the lipids. If you think of like what you do to butter, if you put it in the microwave or in a frying pan, your cell membranes will melt. All right, let's move on. So, measles is a serious disease. A person can die from measles. Table 3 shows the number of medically confirmed cases of measles in England and Wales between 2012 and 2015. Here's the year 2012. There's the case of measles, then 13, then 14, then 15. In your head, what's the pattern? Uh, 5.1 suggests one reason why the actual number of cases of measles in England and Wales might be higher than shown. This is the number of medically confirmed cases of measles. So why might it actually be higher than that? We might say, well, some people don't go to the doctor. Some people do not go. So there's lots of reasons for this kind of answer. It's like any sensible answer. Some people may not go to the doctors. Um, some people are not registered at a doctor's. I mean, that's kind of the same thing. But yeah, some people aren't registered at the doctor's, so they don't report it. Um, Sometimes misdiagnosis happens, so they don't even know. And then, yeah, there we go. That's good enough, isn't it? Lovely. Some people might be on holiday when they get measles and it doesn't get registered. Okay, 5.2, calculate the percentage decrease in the number of medically confirmed cases of measles 2012 to 2015. So what we want to do here, when you want to do percentage change, this is a very, as I say, you probably will never have a set of GCSE science exams where this doesn't come up in one of the papers. They will get you to calculate percentage change. So what you want to do is difference, and this is the equation for percentage change. You want to do the difference over the original and then times 100, and that will give you the percentage change. Now, percentage change is the same as saying percentage increase or decrease. So they might ask you, what's the percentage increase? What's the percentage decrease? Here they've said, what is the percentage decrease? But that is the same as saying percentage change. So we do the difference over the original. So we go 2012. We had the original number was 2030. So that's going at the bottom of my equation. And then in 2015 here, we had 91. So I want to do the difference over the original. So I'm going to go, what do we want to do? 91. What's the difference between 91 and 2030? I'm going to put it in my calculator. So let's go 2030 minus 91 equals 1939. So what I did there is I went 2030 minus 91 to find the difference, and I got 1939, and then we're going to times by 100. And just remember, this is percentage decrease. So I'm going to go 1939 divided by 2030 equals times 100. We've had a 95.52% decrease, 95.52. My calculator says 0.51724138, but I've rounded to two decimal places, 95.52 decrease. So you could write minus 95.52, but it says percentage decrease, so you don't have to. That's how much it's gone down by. And that'll get you 2 marks. Right, let's zing down. Okay. 5.3. One reason for the decrease in the number of cases of measles is that more children were vaccinated against the disease. Vaccinating a large proportion of the population reduces the spread of the measles disease. Explain why. So, if you vaccinate a large, we call this, you might have heard of this word, herd immunity. You can say lots of people are immune, so less chance of disease spreading. So it can't spread even to people who aren't vaccinated. Herd immunity is when you protect the unvaccinated. So the unvaccinated have less chance of catching it. So think in your science class, if 25 out of 30 people in your science class have the vaccine, then only 5 of you can catch it and spread it to the others in that 5. If 29 of you have it, then there's only one person who's unvaccinated in the class. So all those vaccinated people are protecting the person without the vaccine. We call that herd immunity, protecting the herd. Okay, let's zing down. 5.4. Figure 8 shows the concentration of measles antibodies in the blood of a boy. Concentration of measles antibodies in the blood, arbitrary units, 0, 1, 2, 3, 4, 5, 6, 7, 8. The vaccine was injected here. They produced measles antibodies here, then they went back down. Then they got exposed to the actual measles virus, and look at their antibodies they made. Only took like 1 week, and they made like tons, and then it slowly started to come down. Right, we're going to be asked some questions about this graph, so let's have a little look. 6 marks? What? Oh. Explain the differences between the antibody production after the vaccine injection and after exposure to the measles virus. You should include data, right? I'm going to write my answer up here, folks, on the side over here. So after they were injected, what happened with the vaccine? So we want to say the vaccine contained dead or inactive pathogens. So it contained dead or inactive measles pathogens. The white blood cells produced antibodies to bind with measles antigen. So what we find, this explains the increase in antibodies between 1 week and 3 weeks. So after the vaccine, between week 1 and week 3, the person's antibodies have gone up. And that's because their white blood cells are coming into those inactive measles pathogens and they are producing the antibody that will lock on to that pathogen. So just remember the antibody is going to lock on to that measles antigen found on that pathogen. Okay, so we've got an antibody locking on to the antigen. Now, that is going to get you, you've just explained that first section. So that's going to get you some marks. We explain what a vaccine is. We explain how the antibodies have gone up. Then we look at, it looks around 7 weeks. So at 7 weeks, they are exposed to measles. Now, what happens? Memory cells produce a large, look at that, a large amount, that's going to get you a mark, of antibodies. And is it quicker or slower? Look at this line here. Is this line steeper or shallower than this line? It's steeper, which means they are increasing their antibodies over time a lot more. So it's not just larger, but it's faster. So that's two separate marks. Memory cells produce a large amount of antibodies faster. Um, yeah, so faster. By week 8, there are, what do we want to say? 7.2 arbitrary units, so AUs, of antibodies compared to week 3, where we've only got like zero point 0.8 AU antibodies. So I've just done some sort of number comparison. They specifically said use the graph, use data. So that means they want numbers. So what I've said is, look, it goes up to 7.2 after the measles virus, and actually it only got up to 0.8 after the vaccine. So there's a lot more antibodies compared to week 3, where only 0.8 arbitrary units of antibodies. So compared to week 3. So what am I getting marks for here? After 7 weeks, they're exposed to the measles. Memory cells or white blood cells produce a large amount faster. So that's 2 marks, a larger amount and faster amount. By week 8, there are 7.2 AUs, arbitrary units of antibodies, right? So I've given some data. So it's taken 1 week to produce up to 7.2. Compared to week 3, where only 0.8 arbitrary units of antibodies. What I didn't mention in the graph that is also worth mentioning is, look here, it never falls back to 0 again. After the vaccine, some antibodies were left in their blood. So I should have said, which I'm going to write now, between vaccine injection and exposure to measles virus, there are always antibodies in the blood. So before the person was vaccinated, they had no antibodies. There was 0 antibodies. So you might also want to comment there as well and say before the vaccine, they had no antibodies. So I would whack that in as well. So before the vaccine, they had 0 antibodies. So again, that's more marks. So you just say, look, before the vaccine, they had 0 antibodies, and then you're going on to explain all of this. And then in between, their antibody level never goes back down to 0 again. So there's lots and lots of marks to be had here. Cool. Oh, thank God we've done that. I hate 6 markers. Don't you just hate those 6 markers? Right, we are on question 6. So I think this is potentially, yes, it is. It's the last question in the test. Thank for that. The question is about stem cells. Give one place in a plant where stem cells are found. So they're found in the tip of the root and the tip of the shoot. And we call this the merry stem. You could say tip of shoot or root. So it does say give one place. What that means is the examiner will take your first answer. So if I got that right and said the tip of the, if I got it wrong at the start and said tip of the leaf, tip of the shoot, tip of the root, they're going to mark you wrong because they'll take the first word or first thing that you write. And so merry stem or tip of shoot or root. Right. What is the economic use of plant cells? One economic use of stem cells, sorry. To create genetically modified crops, to create a new species of plant? No. To increase variation in plants? No. To produce large numbers of identical plants? Yes. So there is our answer. Right. Let's zing down. Embryonic stem cells divide by mitosis. Figure 9 represents a cell cycle for a human embryonic stem cell. So we got start of the cycle, stage 1, all the way around, stage 2, mitosis, stage 3. The mass of the DNA in the cell at the start of the cycle is 6 picograms. So it's 6 picograms here. Um, A picogram is 10 to the minus 3 nanograms. Convert 6 picograms to grams. Use your answer in standard form. Oh, it's quite hard, that, isn't it? Well, um, if I've got grams and then I've got milligrams and then I've got micrograms and then I've got nanograms and then I've got whatever the hell the sign for picograms is. So a picogram is 10 to the minus 3 nanograms. So um, this is 10 to the minus 3, 10 to the minus 6, 10 to the minus 9. A picogram is 10 to the minus 12 grams. So if I want to convert 6 picograms to grams, I need to times by 1,000 to get them into nanograms, times by 1,000 to get them into micrograms, times by 1,000 to get them into milligrams, and times by 1,000 to get them into grams. So what we're doing here is timesing by 1,000 on the way. No, sorry, we want to divide by 1,000 on the way up. So we've got 6. We're starting with 6 picograms. On the way up the ladder, you divide by 1,000 each time, right? On the way down, you multiply by 1,000. It's kind of counterintuitive. So I want to go 6 divided by 1,000 divided by 1,000 divided by 1,000 divided by 1,000. And I'm going to get 6 times 10 to the minus 12. There we go. I could have just divided by a billion because a picogram is, oh no, is it a billion or a trillionth? That's 1,000, that's a million, that's a billion, it's a trillionth. Oh my days. Woo! Right, that's how many grams 6 picograms are. My brain's melting now. 6.4, the time taken for this complete cell cycle is 15 hours. Calculate how many hours the cell spent in mitosis. Give your answer to 3 significant figures. So the whole thing took 15 hours. Calculate how many hours the cell spent in mitosis. So what you've got to do here is get the old protractor out and measure the size of this. So mitosis is 28 degrees. You don't even need the protractor. What do we know about a circle? A circle is 360 degrees. Mitosis was only 28 degrees of it. So I want to do 28 degrees divided by 360 to find out the proportion of the cell cycle that was mitosis, and then times that by 15 hours, which is the full lot. So we want to do 28 divided by 360, and we get the proportion that only 0.078 of all the time, that's the proportion of time the cell spent in mitosis. We times that by the total time for the whole cycle. So times 15 hours equals 1.16 or 1.17 hours out of 15 were spent in mitosis. And that was worth 3 marks, 3 significant figures. So I want 1.17 hours. And that is worth 2 marks. Bish bash bosh. Right, I'm right near the end of this paper. I think this may be the end. Ah, it's a 5 marker. Is that the end? No, there's a little bit more. 6.6. Describe what happens in each of the 3 stages of the cell cycle. So we've got 3 stages of the cell cycle. So in the first stage, new proteins are made. The cell grows. Oh, for God, Lord of mercy. Right. The cell grows. It would help if I actually used commas or full stops rather than just smashing it all out. Right. First stage, new proteins are made. The cell grows. Oh my days. New organelles. You probably heard of new organelles, but you can just say new cell structures. E.g., new ribosomes. You're going to split in two. Imagine you were going to split in two. You'd need 2 hearts, 4 kidneys, 4 lungs, because you're going to split in two. So the cell has to make new ribosomes, new mitochondria. Before it splits, it's got to double everything. So that first stage is really long because it has to make protein, it has to make ribosomes, mitochondria. It's got to double everything. And then it also needs to double the DNA or double the genetic code, double the chromosomes. So we're going to double the chromosomes. And that's all in the first stage. Then, oh, yay, in the second stage, we can say chromosomes line up as X shapes on middle of cell. They are pulled apart, one arm, oh geez, of the X going to each end or pole of the cell. And that is in the second stage, and we call the second stage mitosis. Now, in the third stage, Oh. In the third stage, the cytoplasm divides and the cell, we call this cytokinesis, splits into two identical daughter, oh, daughter cells. And there's your cell cycle for you. Wunderbar. That means wonderful in German, if you're wondering. Right, this is the last bit, last bit, last bit, last bit. Thank the Lord. 6.6 figure 10 shows how the embryonic stem cells are produced in therapeutic cloning for use in patients. So we take a body cell from the patient, you're sick with something, right? We take your cell, we chuck your cell away. We don't want all your cytoplasm and your ribosomes and your mitochondria. We don't need that. We just want the nucleus with your DNA of the patient. That's all we want. Then we're going to get an empty egg cell. We're going to trick it into thinking a sperm has gone into it. We're going to make it think it's been fertilized. So we chuck away all the DNA from the egg cell and we insert the nucleus from the patient cell into this empty egg cell. We zap it with a little electricity. It thinks a sperm and egg have joined when really you've just put the full DNA of an adult into that egg cell. It will then start to divide and differentiate and clone itself by mitosis and specialize, and it becomes an embryo. They're all identical still then, but then some of the stem cells are cultured, some cells are not used. Give two advantages and disadvantages of therapeutic cloning in medical treatments. So the advantage is that now any donor organs or anything grown from it is going to have less chance of rejection. So less chance of rejection as antigens are the patient. So the main advantage is if you give someone a donor organ, and I could say the second advantage is you can grow organs for donating. Um, but there's obviously going to be less chance of rejection because it's got the patient's own antigens. So we can also say, ba, 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 oh my gosh. We can also say, um, organs save lives. You know, you're going to save the life of people who need a new heart or a new kidney, and there aren't any kidneys or hearts about. So you're going to save their lives, and that is a really good thing. Can cure medical conditions, can cure paralysis, maybe, can cure medical. We can't cure paralysis yet, but we can cure medical conditions, certain diseases, and things like that. So lots and lots of different advantages there we could give. The main one, of course, is that you reduce the chance of rejection when doing organ donation. Now, what are the disadvantages? Right, what happened to some of the cells that are not used? Some embryos are destroyed. So you've destroyed potential life. So potential life destroyed. So some people will call you a murderer for that, although, you know, that's kind of a bit extreme, isn't it? But yeah, some embryos are destroyed in the process if they're unwanted or unkept. There might not be any egg donors. So you still need someone to donate an egg, and there might not be any. And one of the big things is viruses. Virus could be in the donor cell. So it means like you could have COVID inside the donor cell or the flu or the HIV virus hiding in that cell. When you mix it with the patient cell and put it back in the patient, they've now got that virus. So you might transfer a virus to someone. So those are some of the disadvantages and some of the advantages. All right, guys, I hope that helped. I'm going to try and smash out a couple of these every week to help you get to your exams and smash those exams. Remember, if you are a separate scientist or a combined scientist, you should do both. You should have a look at the triple papers and at the combined science papers because 80% of the content is the same. So you're going to get some good practice. And remember, the examiners may take questions they've asked in combined science and they go, oh, we haven't asked this stem cell question yet in triple, and they stick it in the triple biology. So you've got practice if you did the combined science paper. You also, vice versa, you might get something from one of the triple papers that is part of the content for combined science that they've not asked yet, and it gives you practice on those topics that you haven't come up yet in combined science. Anyway, enough of my waffle. Remember, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll see you in the next video.
You're still here? It's over.